Toxicological approaches have changed little in the past few decades, but they are now on the cusp of an omics revolution. Among the greatest challenges in toxicology are elucidating deeper insight into the mechanistic causes of toxicity. How do toxicants interfere with the body's complex network of biological pathways to cause disease? To answer this, new technological approaches, such as those enabled by Agilent's metabolomics, transcriptomics, and bioinformatics tools, are being used to understand toxicological mechanisms. Please share a few moments with us in order to learn about how Agilent is enabling toxicologists with new tools that help address these challenges. Professor Hartung promotes the view that the future of toxicological testing will be increasingly based on proteomic, transcriptomics, and metabolomics assays on cell lines wherein researchers will look for activation of predefined pathways of toxicity.
We are seeing that we have developed a very precautionary type of toxicology, which might make it difficult to bring something like aspirin to the market today. We are seeing that animal use is considered more and more critical by a general public, and we are seeing that new products like biologicals, nanoparticles, cell therapies are requesting different type of approaches for safety assessments.
Professor Hartung promotes the view that the future of toxicological testing will be increasingly based upon batteries of assays carried out using cell lines wherein researchers will look at activation of predefined pathways of toxicity.
There's probably a couple of hundred ways to harm a cell, but we believe it is not an endless number. For the very simple reason that the number of critical infrastructures in a cell is limited. If we would be able to annotate these pathways, to describe them on a molecular level, we might be able to come up with something like the human toxome as the entirety of pathways of toxicity. And we could start annotating these pathways to cells, to different cell types, and understand why a hepatocyte is vulnerable to a toxicant while a cardiomyocyte is not. We could annotate them to classes of toxicants, understanding what are the critical perturbations which lead to a certain phenotype or hazard manifestation.
Hartung believes that up to now, a key change in making sense of the measurement of the up and down regulation of genes, proteins, and metabolites within biological pathways has been the inability to integrate this data with existing knowledge in order to be able to fully visualize, for example, the way in which a drug compound might adversely affect a biological pathway. Agilent's recently launched GeneSpring 12 software addresses this challenge, enabling toxicologists to gain a greater understanding of the mechanisms of toxicity, as well as the more accurate assessment and prediction of toxicological endpoints that could result in, for example, injury to the liver or the development of a human embryo. Stemina Biomarker Discovery is applying state-of-the-art metabolomics to the development of new in vitro toxicity testing methods based on human embryonic stem cells using Agilent LC-MS technology.
I'm Beth Donnelly, and I'm CEO of Stemina Biomarker Discovery. Stemina was founded in 2007 to commercialize a technology that came from the University of Wisconsin, from the laboratory of Dr. Gabriela Cesar. And the idea that Gabby had was a really fundamental one for toxicology. It was, could we build a better way of understanding human developmental toxicology by using human embryonic stem cells and a science called metabolomics? So in our work here at Stemina, we do just that. We have an assay called DevTox that's used to predict whether or not drugs and chemicals will cause birth defects if a woman is exposed during pregnancy. We do that through using human embryonic stem cells, which come from an embryo, so they're able to recapitulate every cell in the body. And we can study in a way we never could before what goes wrong when that embryo intersects a compound or a drug that might cause birth defects. And we do that using Agilent mass spec equipment and a science called metabolomics. So metabolomics is the study of our metabolism, similar to the way genomics is the study of your genes. However, it's the closest omics to your phenotype. So in other words, we can understand what's happening in real time in a cellular system or in a patient by studying the small molecules that they secrete in response to a toxic substance or a disease. There are a number of other omics, genomics, transcriptomics, and proteomics, which together comprise the systems biology approach for understanding diseases or also exposure to toxic substances, for example. Agilent also provides technology that relates to the other omics. And so our hope is during our collaboration with Agilent, we'll be able to add a metabolomics component to the overall systems biology technology offerings that they have. With Agilent, we use the mass spectrometers to study the metabolomics endpoints, but we also have a collaboration with Agilent under which we test new software such as Mass Hunter and Find by Formula to try to establish a relationship from our metabolomic endpoints into the entire omics approach for a more systems biology understanding of the particular question we're studying. As we develop our toxicology assays, we're also working in the area of cardiotox and neurotox, using cells that are made from stem cells, which are physiologically relevant, and again, the same metabolomic endpoints. Agilent has been very supportive of our efforts during our collaboration by providing access to new software, which helps us to analyze the data that we create during these studies so that we can look at new ways of sort of examining the blood and coming up with new biomarkers.
Curious to learn more? Visit our website to download your copy of Agilent's Pathways to Insight and Food for Thought on Systems Toxicology. And sign up for our upcoming seminar session.